If we can get everybody seated, we'll begin soon. I'm Robert Bjork, chair of the Jeffrey Lecture Committee, and it's my great pleasure on behalf of the Department of Psychology and our chair, Bruce Baker, to invite all of you to the 12th annual Jeffrey Lecture. Before I go further, there's a person I just have to thank who's in the back there somewhere. Deanna Evans, stand up. Deanna has arranged every detail for everything. And yes. So developments in cognitive and more broadly behavioral neuroscience have energized psychology and really all of the behavioral sciences. They've intrigued the public and national media and have drawn truly gifted researchers and students to our field. This lecture series, across its 12 years, has brought a who's who of cognitive and behavioral neuroscientists to this campus. And with today's speaker, B.J. Casey, who's director of the Sackler Institute for Developmental Psychobiology and is a professor of developmental psychobiology at the Weill Medical College of Cornell University, we continue that tradition. Before I ask Nim Tottenham, who worked with Dr. Casey as a postdoctoral fellow, is now a professor in our developmental psychology program, to introduce Dr. Casey, I want to again thank Professors Emeritus Wendell Jeffrey and Bernice Wenzel, not only for funding this lecture series, but having the idea for it in the first place. Jeff and Bernice, after coming to UCLA in 1955, have been amazing assets to this university, not only during their time as professors at UCLA, but continuing on since their retirements in 1990 and 1989. Jeff built our developmental psychology program, and he served as editor of all three of the most prominent developmental psychology journals of his time, including as founding editor of the Journal of Experimental Child Psychology, and his honors include a unique award from the American Psychological Association for his remarkable record of mentoring developmental psychologists who are now leaders in the field. Bernice, too, has had a wonderful career at UCLA, both pre-retirement and post-retirement, and among her other honors, has been named Emeritus of the Year by the UCLA Emeriti Association. I want again to thank Jeff and Bernice for their generosity and devotion to UCLA and to the department in particular. Among other ways they've supported our department is they have endowed the Wendell Jeffrey and Bernice Wenzel Term Chair in Behavioral Neuroscience, which is a critical tool for the department in supporting and retaining outstanding young researchers. Jeff, Bernice, please accept our gratitude. So on with things. Nim, would you please introduce our speaker?
So it is my extreme honor and pleasure to introduce our speaker for the 12th Jeffrey Lecture on Cognitive Neuroscience, Dr. B.J. Casey. Dr. Casey is the director of the Sackler Institute for Developmental Psychobiology in the Department of Psychiatry at Weill Cornell Medical College in New York City. Since 1998, and under Dr. Casey's directorship for the past decade, the Sackler Institute has gained an international reputation for research and training using the techniques of brain imaging, human genetics, electrophysiology, and behavioral methods to study typical and atypical human brain development. Dr. Casey was selected for this honor today because she is one of the greats, one of those pioneering thinkers who has had a tremendous and widespread impact on the shape of neuroscience today. Because of her work and ideas, we have a much clearer picture of normal human brain development. Dr. Casey was the first to publish a scientific study using functional magnetic resonance imaging, or fMRI, to examine brain development in the healthy and awake child. And today, there are over 800 papers on human brain development using fMRI. Before Dr. Casey's work using fMRI, our understanding of brain function was limited to studies that relied on more invasive measures and thus could not be used on healthy, typically developing children. With the advent of fMRI, Dr. Casey was, for the first time in history, able to examine the dynamic changes in brain architecture that construct changes in behavioral development. So when scientists today say things like, we know that the prefrontal cortex is a late developing structure in humans, which is one of those facts about brain development that has become so fundamental, it is almost said with a casual tone. We are largely referencing the early pioneering work of Dr. Casey. Since then, her work has had large and significant impact on the areas of adolescent neurobiology, ADHD, and other disorders related to attention, autism, early life stress, and emotional learning. She is one of the few who has truly defined what it means to perform translational science. She combines the use of fMRI with animal models and clinical populations to better delineate the genetic and neural mechanisms that contribute to pathological behavior. She has always been a trailblazer with the foresight to predict how science should move forward, and as a consequence, she has worked very closely with the NIH on their blueprint initiatives. She also contributes much of her time to serving on grant review panels every year for the past 2 decades for NIH, NSF, and several other foundations. She has published over 100 peer-reviewed scientific papers, plus an additional 34 books, chapters, and reviews. She is a recipient of numerous awards, including the John Merck Scholar Award in the Biology of Developmental Disabilities, the Charles Dana Clinical Hypotheses and Neuroscience Research Award, and been named Alumna of the Year Award by her alma mater, University of South Carolina, twice. She has been awarded numerous grants through the NIH and NSF, as well as receiving plenty of grant support from various private foundations. She is currently the principal investigator of a center grant, a P50 grant, examining the effects of BDNF genotype and stress on learning and development. This is an interdisciplinary center that uses a true translational approach to test gene-environment interactions on neural substrates of learning across development in humans and mice. In addition to her large and impactful research career, BJ is truly a dedicated teacher. In addition to the large number of students that have been lucky enough to train under her, BJ has always placed training of students from numerous labs, both from across the nation and internationally, as a top priority. Every summer for the past 11 years, BJ has organized the Sackler John Merck Fund Summer Institute, Biology of Developmental Disabilities. This week-long course features expert speakers whose work focuses on the organization and development of brain systems mediating learning, emotion, and reward processing in their relation to developmental and psychiatric disorders. It's a once-in-a-lifetime experience for a student of neuroscience that includes not only intellectual mind-blowing, but also tons and tons of fun and laughter that routinely and naturally accompany BJ wherever she goes. I was lucky enough to be a student in BJ's lab and attend these summer camps. I would like to add a little personal note in here so that the audience can get a true and full picture of BJ. I should first mention that BJ is extremely humble, perhaps one of the most humble people I know. Therefore, I'm going to say things that might make her blush, but they are truly from the heart. She created a lab and program of research that is priceless, full of hard work and full of laughs. Although she is a force often associated with the technique of fMRI, she has always emphasized the importance of understanding cognitive behavior first and how our understanding of behavior might be constrained with neuroimaging techniques. She has made me appreciate that the goal of a developmental neuropsychologist is not simply to study the brains of children to see if their brains act in the same or different way as adults, but rather to study the construction of a neural process across ages. She transforms the way you think to make you think better and be a faster thinker. And through my eyes, she was always the fastest thinker in the room. And that in and of itself makes you want to think about things the way she does. It is a fact that I would not be where I am today were it not for my time with her. And I also know that all of her other students feel the same way. She has had the most direct impact on who I am as a scientist, and I'm not sure if it is possible to appropriately thank someone for something like that. But I am and will forever be grateful to her for that influence. So with that, I will complete my introduction so that Dr. Casey can begin her talk. So please help me by giving her a very warm welcome to UCLA and the Jeffrey Lecture on Cognitive Neuroscience. Thank you.
So I think, Adriana, you would understand the statement if I said I feel like I was just attending my wake, or the wake that you would like to attend. It really is such a privilege to be here. It's lovely. It's a really honor to have been chosen to give the Jeffrey Lecture. And I have to be really honest in saying that I visited a number of cities in the world, and I do live in that wonderful city of New York, right smack dab in Manhattan. But it's only when I'm returning from a trip to LA when I typically see that beautiful skyline of Manhattan and get this just moving, this exhilaration, that when I come back from LA, I actually get this pang or this yearning. And some of you will go, well, of course, the weather's a lot better here. But it's also because you have such a wonderful scientific community and family here at UCLA. And it's really humbling to give this talk amidst so many people who have been so influential in my work that, again, thank you very much and for that awesome introduction. So the most important slide you can ever present in a lecture is a slide that acknowledges all the people who come together to bring the work that you present together in that format. And I'm highlighting, of course, two of my own students now with very strong independent careers of their own, Dr. Tottenham and Dr. Galvan. And in addition, all the fellows who each little idea has contributed to the ideas in the group. No one knows how things are going to evolve in terms of trying to understand that wonderful final frontier of the brain, but it has been a wonderful agenda. Do I need to move this? Just a second. Is that cool? Awesome. Okay, thank you. So now, I'm about to deliver statistics to you that every time I recite them, and it's been over 100 times, I get a visceral reaction because I am a parent of an adolescent. 17-year-old, so he's slowly moving in the direction of adulthood. But it's very alarming to think that the leading causes of death in our youth, the three leading causes, are ones in which the teenager typically puts themselves in harm's way. So it's accidental injury, it's suicide, and then in some cases homicide. This is also a period of time during which we're seeing peaks in the diagnosis of affective anxiety and mood disorders as well as substance abuse. And if that weren't enough, this is also a time when we see spikes in criminal activity across this age range of roughly 13 to 17 years of age. So perhaps it's not surprising that when individuals talk about adolescence, they want to psychopathologize this developmental period. They want to talk about how the individual is impaired or how the individual has deficits. Many times they suggest that this impaired thinking is due to suboptimal decision making and that in fact adolescents can't make rational decisions. And then the delayed development of the prefrontal cortex is what they then assume is underlying these problems with the behavior. But that doesn't logically follow when we think about children who have an even less developed prefrontal cortex, who should be showing the same spike, but they don't. Now, the evidence for a protracted development of the prefrontal cortex comes from human imaging, but there's a long history in the animal research, non-human primate, and also post-mortem studies. So we understand why that gave individuals some traction in trying to understand what might be going on in the adolescent. But really, the passionate question that we've had is, why do adolescents engage in the behavior that they do? And why would the brain be programmed in such a way to help facilitate them engaging in some of that behavior? And I really just jumped right on board when I was recently contacted by David Dobbs, who wrote a beautiful story, actually referred to as A Beautiful Brain, in National Geographic. Because David has a teenager himself and had recently had an episode with David. It's a lovely story. I would recommend you reading it and hearing exactly the way David tells it in terms of a speeding incident and his son being picked up by the police. Otherwise, a very good kid and thinking very normally as a normal phase of development. But he wanted to treat this time and this period and write the article not about psychopathologizing the adolescent, but about how this experimentation and what they were doing is really a natural part of development that allows them to proceed to the next independent stage. And I think this is probably a good time for me to introduce the definition for what we mean by adolescence. So it's a developmental transition from dependence on the parent to relative independence with onset at puberty. Offset, well, if it's relative independence from parent, that could go on for a long time. But it's characterized by this increasing sort of experimentation, a heightened sensitivity to social pressures, and also what we've seen across species is this novelty seeking during this period of time. And so what we've tried to provide to understanding the adolescent brain and thinking of it more as an adaptive phase and how this behavior in the context of adolescence and what they're trying to do in terms of gain independence, experiment, and understand the social boundaries of their behavior, is to think about it from a neurodevelopmental progression. So this is going to take into consideration what's going on not only during this period, but what precedes it and what follows it. This is actually a blueprint initiative in thinking of transitions into and out of developmental windows. Without that, it makes it very difficult to say anything about a particular developmental period. It's also important to track and delineate the developmental trajectories, both in terms of function and structure. And so there are 2 points I want to make here. Many times people come to the Sackler Institute and I ask them, wouldn't they share with us their latest findings? And they say, no, I'm just doing behavioral research. And you guys do imaging, so you wouldn't find it interesting at all. Well, that's so far from the truth, it hurts. So we're interested in behavior, and we're using imaging to try to constrain our theoretical views about behavioral phenomena that we're seeing. Likewise, when we do imaging studies, and when anyone does, it's nice to be able to constrain your interpretation of the patterns that you're seeing with the brain imaging with the behavioral changes that you're seeing simultaneously. So we'll come back to that point as well. And then third, since behavior is so important, we really need to shift, and I think this is the neurodevelopmental perspective. If you're trying to link the behavior to the biology, then we have to simplify the behavioral measures that we're using enough that they can be linked to the underlying neurobiology. And in the case of adolescence, many times we use almost synonymously the adolescent is impulsive and sensation seeking, or impulsive and risk taking. And these are really different constructs. So what I'd like to do today is provide empirical evidence for dissociating these constructs, how they have different developmental trajectories, different underlying neurobiology, and looking at that from more of a transitional phase. Now, we've done a number, as Nim mentioned, we've done a number of across-species studies too. And while I'm here, I almost got asked to give 4. I did get asked to give 4 talks, but I'm giving 3, and I'm going to save the animal work for next Tuesday. So I apologize to anyone that wanted to hear about the parallel mouse and human studies across development. It really is cool, and I'll be glad to send you the slides if you can't make that talk. I think it's also a really good idea when you give a talk at this time in the afternoon to give take-home points, because take-home points, when you hear them in the talk, are like cues of the reward to come, which means the talk's going to be over, and you can have a reception, and you can have the conversation with each other about pros and cons or interesting ideas or not so interesting ideas. So, so let's just go straight to the take-home points, and then I'll unravel those over the course of the lecture today. So the first is, unlike what many of the misconceptions about adolescence are sort of out there in the lay public and also by the media often, and some of our colleagues, Adolescents are quite capable of making good decisions. The problem is many of their decisions that put them in harm's way are in the heat of the moment and they're immediate. And so I want to try to spend some time about how they perform in the immediate and when you give them a little bit of time to cool off. And then the second is the very same circuitry that might put them at risk for making a bad decision may also be very important in terms of thoughtful choices that are appropriate. So first, let's just start with what we know about this regional changes in the brain that might map on to the behavioral changes that we see during adolescence. It's one of those number one points from the neurodevelopmental perspective. And this just highlights across the lifespan, although we're really focusing on adolescence today, that there are a lot of changes. There's both regressive and progressive. We have pruning, we have myelination going on, and really it's a combination of those with experience across development that are fine-tuning the circuitry. And I think the best evidence from the human imaging comes from work that was also in collaboration with Paul Thompson here, is work by Jay Gee, Judy Rappaport, and of course Gautier, and showing, that movie might have gone too quickly, so let's just look at a static image, showing that cortical, a measure of brain development, cortical thickness, actually peaks in regions that are associated with primary motor and visual capabilities. That's indicated in blue when it reached peak adult levels. And those areas, an association cortex looking down again on top of the brain, that take longer to develop are in areas like the prefrontal cortex and of course also language regions. For most of the lecture today, in fact, all of it, I'll focus on the prefrontal cortex. Elizabeth Sowell, when she was training with Terry Jernigan, I think she's recently moved on, but was very productive here at UCLA in this strong community of scientists, actually also showed that, we can't just be these cortical chauvinists. And in fact, when you think about adolescent behavior, it really is probably these deep structures of the brain that you're seeing in this glass brain and little globules of green and blue. These areas are still changing just in structure from late childhood into adulthood, just as regions in the prefrontal cortex are. So many of these regions are those that are part of the limbic subcortical systems, and they're heavily involved in desire and fight and flight. And on Tuesday, I'll talk a little bit more about emotion regulation. Today, I'll talk more about incentives, and tomorrow, I will talk about risk-taking. Yeah. So this work has sort of led to working particularly with Adriana Galvan to this imbalance model. Several individuals had put it, a model like this on board in terms of how there's a tension between prefrontal and sort of limbic. For any Star Trek fans, I guess I can do this in LA, it's like Dr. Spock and Captain Kirk, and so there can be a tension there, but we're not trying to say that these are orthogonal systems. We're trying to talk about this in terms of circuitry and how these pathways, there's not been enough experience or over the course of development to actually fine-tune the projections that will help suppress the emotional response. And so we've spent a lot of time trying to find ways, let's see if this is going to work, trying to find ways in which these deep structures, we can sort of isolate those and then look at them in combination with prefrontal regions that project to them, and then most importantly, try to develop tasks that we can take into the scanner environment that tap into both. Because that's when we think we're going to see a tension during adolescence. And so hopefully I can give you some empirical examples of that. Now this was a little hiccup in a movie. It's not as suave as most Hollywood movies. So why don't I just again show you a static image and again work with Adriana Galvan in forming this model. We were very displeased in thinking that the prefrontal cortex was going to explain adolescent behavior. So we were trying to get a handle on how to local connections in these subcortical regions, how might they then bias cortical projections to these more local subcortical regions. And the imbalance model goes like this. You don't see it in children because both of the circuitry is still developing, it's less mature, In adulthood, you don't see it as much because at this time, the system is fine-tuned and mature, but it's during this stage when you're going to get interference or there might be an imbalance. And there are a number of ways that we've looked in our lab in terms of special populations with various psychiatric disorders that exacerbate this imbalance even more. So far what I've done is sort of lay the groundwork for showing that there are regional differences in the brain in structure. But what we want to do, remember, is talk about function. And I always say that your at least task-based functional imaging is only going to be as good as your task. And if your task doesn't measure what you're trying to get at, your imaging isn't going to tell you a whole lot. And this is really important when you're trying to look at psychopathologies and trying to come up with an assay that gets at the core problem that that individual is having. So when we think about what's going on during adolescence and across development, a hallmark of the changes that we see are an ability to suppress inappropriate thoughts and actions in favor of appropriate ones. And this gets particularly difficult, even for those of us as adults, in very heated situations. Now, what I'd like to do is show you some of the experimental tasks that we've used in the lab to get at these simple aspects. Remember, I said we're trying to look at very simple behaviors so that we can more easily link that to the underlying biology. And we also want to try to tease apart impulsivity from sensation-seeking or risk-taking. So first, I just want to start with how do we measure impulse control in the laboratory? And because it's about 7:30 Eastern Standard Time, I thought I would let you do a little bit of the work here. And also because it's the end of the long day, I thought we might be able to engage you by performing this impulse control task. Now, we have a number of versions of this. This is one of my favorite because before I moved to New York City, I lived on a small farm in North Carolina. During that lifetime experience, I learned that eggplants do not grow out of the ground, but that root vegetables do. However, when you work in New York City, many of your research staff and fellows are actually not country folk, but city folk. And so I didn't want them to change the stimuli because it was very compelling, and it always makes me giggle when I see it. But we've used a number of other stimuli that are popular. Right now, Spider-Man is back in because we have the whole Broadway show where there's always someone getting hurt or something is going on these days in Manhattan. And so when I had the audience do these tasks, like whenever Spider-Man is there, you don't want to press, you want to press the button, but when the Green Goblin's there, you don't want to press. And so one time I had the Department of Psychiatry, I forget which institution, one of the Ivies, I guess, and I was having them perform the task, and they got really, really excited, and they were giving these yes responses really loud and really quick. And I began to notice that people were beginning to come from the outside into the auditorium as they were hearing, yes, yes, yes. So for today's task, instead of saying yes, I'm going to have you say whack. And maybe, you know, I was thinking that's appropriate for whack-a-mole, but maybe, okay, I'll just take a step back. So you know I know, okay. So, but the task is to sort of let go of your frustrations. If you've ever actually played this task, not in our laboratory because it's not as much fun as the one where you're just kind of beating the gopher or whoever on the head. In this one, whenever you see this little sneaky mole that's digging up the garden, I want you to just say whack. And whenever you see this eggplant magically emerging out of the ground, don't say anything at all. Okay? So, are you ready? Yes. Okay. And we need enthusiasm. So, one of some things that we notice is there's a lot of individual variability in performance. You don't just have to be a young child or an adolescent to have difficulty on this task. And if you'll pardon the Irish expression, I really feel like the individual differences many times are where the meat and potatoes are, particularly when you're thinking about psychopathology and wanting to know what's going on with adults under this line and above that line, although all of these are healthy individuals, just as all of you are healthy individuals. We tend to have a few little false alarms there. But do note that overall, the general pattern is sort of a linear increase in your ability to perform the task. I plotted this as a function of D prime only because it takes into consideration both how well you know what the target is, but also how well you know what the non-target is. So it takes into consideration false alarms and hit rate. But if we plot it like you typically do in developmental studies, you see this nice, neat, linear decrease in the proportion of false alarms as a function of age. And if we take this task into the scanner and we look at what part of the brain is correlating with how well you perform this task, because when we do imaging studies, sometimes the whole bloody brain will light up. But what you want to know are what are the regions that are really specific to how well they perform the task itself. And we see time and time again, Sarah Durston has shown this in collaboration with myself and in elegant independent work. Evelyn Kroon in the Netherlands as well has shown the same sort of right inferior frontal gyrus coming up repeatedly whenever you're having to suppress behavioral action in these impulse control tasks. And it's also correlated with just how well individuals do on the task. And you're going to notice that, I don't know if you can see it from the back, but these are more adolescents and children, and the adults are really hanging out here because their accuracy is very high, as you see from the previous slide with the small number of false alarms. But what do you notice about the slide in terms of the adolescents? Anything special? They're on this trajectory. But are we seeing spikes like we see in criminal activity? Are we seeing anything that gives us traction with regard to the health statistics that I reported? Not really anything. It's a linear change. They're improving with age. So what we now want to do is see, earlier we were talking about adolescents when they're engaging in a decision, when they have to make one, it's in the heat of the moment, and it's almost immediate. So now what we want to consider is how are we changing in terms of our representation of incentives and how they bias behavior? So how are incentives represented in the brain across development? And then, once we sort of look at that, then we can combine that in combination with the impulse control task. Now there's a huge, elegant literature showing that cues that actually predict rewards and good outcomes, that we tend to approach them. We actually are faster to them. One nice example of this that Adriana published about 5 or 6 years ago was in this wonderful pirate task. This is building on Wolfram Schultz's work on dopamine firing because she was interested in really how does the pattern that we see in brain imaging have anything to do with reward in any of the non-human primate or animal studies that have been done. So it was great for her to use a task that was almost identical to what had been done in the animal work. But the pirate task had 3 pirates. There was one, I think, with a parrot. There was one maybe with a patch. And then there was one with a hook. And the pirate's favorite letter is? Yeah, R, yeah, sorry. So, and then each one of those distinct stimuli were associated with a distinct amount of reward. Notice that there are not numbers up here because children and adolescents will turn this into an arithmetic task to find out how much they're going to gain or earn by the end of the task. And you don't want them to do that. You just want to see, in general, that there are incentives associated with those cues. And even without them being consciously aware of the association between each of these pirates with a different amount of reward, you observe much faster reaction times to the one that is associated with the largest reward or predicts that. And one thing we really weren't expecting is you're still getting rewarded with a single coin to this stimulus, but you see significantly slower responses to this one. So this is suggesting how cues out in the world that have been associated with incentives at some point in time can very easily bias our behavior, and we're not even aware that they're doing that. This is also really at the very basis of a number of psychiatric disorders as well. But now what we want to do is is look to see what areas in the brain are associated with reward, but more importantly, how do they vary across development from childhood to adolescence to adulthood. And two areas that come up time and time again, one is the orbital frontal cortex, but the area where we continue to see developmental differences is this area in the ventral striatum that's very important in learning about cues that predict outcome and also detecting when what you're expecting to occur does not. And so I think this was one of the first reports developmentally showing that in the ventral striatum there was a heightened response in the adolescents relative to both children and adults. And there was another just elegant paper that was published by Jessica Cohen when she was a graduate student. I think she's a postdoc at Berkeley now. She was working with Russ Poldrack, but Susan was on that paper, numerous, Fred, Barbara, Bob, and Bob. But basically showing that adolescents had this heightened positive prediction error. So they learned about cues in a heightened, enhanced way that led to positive outcomes. Other labs that have showed this blip, which again is to take note because this goes with the blip or the spike that we see in some of those health statistics I reported, is work in the Netherlands by Evelyn Kroon, where she showed enhanced ventral striatum at receipt of money as opposed to children and adults. Another person who's been doing absolutely seminal work in adolescent brain development, Bea Luna at Pittsburgh, shows that in anticipation of a reward, you actually activate this more in adolescents, relative adults, same region. And then Larry Steinberg, who has spent part of his sabbatical here in LA, has shown that when you engage in a risky choice in the presence of a peer, you activate this region more if you're an adolescent, but not if you're an adult. Bringing in that whole peer influence. In fact, is as basic as if you're presented with a positive social cue, like a smiling, happy face from the NIM stem that Dr. Tottenham has developed and are being used worldwide, such as a happy face, you tell someone not to press to that happy face, and we see elevated activity in the ventral striatum in adolescents relative to children and adults. But earlier on, I said one of the things that we needed to do with our imaging data was to constrain that by what's going on behaviorally during this period too. So if we look at the behavioral performance on that whack-the-mole task, and now what you've done is instructed the individual, don't press to the happy face, and they're embedded in neutral ones. Look at what we see in the behavior. So relative to children and adults, the adolescents are approaching, making many more false alarms to that happy face, as if they can't stop themselves. And they all have sort of an equal level of hits here. Now let's bring this full circle because we're trying not to be phrenologists here and say there's this opposing or orthogonal region and this region. We're talking about a circuit. And Leah Somerville, who's Harvard-bound for a faculty position next year, she's been working on this project with us along with Todd Hare. And she did functional connectivity analysis to try to understand this behavioral pattern with the imaging data that I showed you before, here with the ventral striatum, and looking at seed points. And what she shows is, let me try to unpack this diagram for you. This top level is the projection zone in the prefrontal cortex for this task. Below is the dorsal striatum. It's this circuit that helps you do a go, no-go task. The ventral striatum is part of another circuit that can bias your performance on the go, no-go task. And where we're seeing a significant coupling is only in the adolescence. It's emerging during childhood, and we're not seeing it in adulthood. Probably or potentially because by the time you're adult, you have this nice fine-tuning and you know how to suppress appetitive cues. If the task is to stop, for the most part you do, although a few of us sometimes have issues. And I'll talk about those interesting individual differences tomorrow as part of a delay of gratification study we've recently published. So this gives us a little bit more traction in terms of the circuit and moving away from just phrenology and how subcortical systems may be talking to one another. There's a lot of resting state imaging data. I don't some of you are aware of that's not task-based. But you get these strong projections from the dorsal and ventral striatum. You can see those with the resting state. And I think that's nice in how it corroborates some of these findings. So now, remember those anchor points for the take-home messages. One of the take-home messages is that the very same systems that seem to get adolescents into trouble also seem to be the ones that help them make careful or thoughtful judgments. And I want to just remind you of Bea-Luna's findings. This is a busy slide, if you just focus on the right, this was showing that adolescents activate the ventral striatum more when they're anticipating their task performance being rewarded. On the left, it's showing on an impulse control task that they significantly improve when they think they're going to be rewarded relative to when they're not. And adults show a little bit of improvement too. Now, what is this impulse control task? And even though the ventral striatum is active, what is it active on? What it's doing is modulating the frontal striatal loop that's responsible for impulse control. But this is a slightly different impulse control task because it's looking at eye movements. So basically, she does something very tricky to her participants, and children find it almost so difficult to do, it's hard to get enough data. But she has them perform what's referred to as the anti-saccade task. So there's a little light over on one side, but that means you have to look to the other side. So let's make sure that everybody's still awake here. And we're going to engage in this task. When I move my arm out like that, I want you, since I can't see your eyes from where you are, if you make a head turn. And when I move my arm like that, then you're supposed to look in the opposite direction, right? So you're going to look that way. You got it? So let's see how well you can control your impulses on this task. Whoops, I'm seeing a few of you doing that. I think it's more fun being up front than it is. Is this air traffic control? Or is it a cheerleader for the Giants? I'm so sorry for any 49er fans. Okay, sorry, that was bad. I may have just lost you there. Now, let's just come back though. So this is the task. But when you're performing it, instead of you constantly being prepared to make a motor response, which involves slightly different ventral prefrontal circuitry, this is about eye movements. And so that involves the frontal eye fields and the ocular motor circuit and modulating it. But there's been some criticism of this task because notice in the adults, they're improving, but to some extent, they're almost at ceiling on this task. So it could have been that they were going to improve just as much. So what we wanted to do is to try to use a task where we could equate performance across ages. And two, overall, the adolescent literature and the findings I presented you so far have been criticized in part because it's been suggested that faces and peer expressions and monetary rewards are more salient to adolescents than to children and adults. So we wanted to try to equate reward and not use anything that they would think would be a gain in society, since they are trying to get to the next level of independence in society. So rather than using money or facial expressions or interactions with peers, we just simply said points. We're just going to use points. So it's going to be 1 point in one condition and 5 points in the other. And let's see how you perform. And the task that we used is this beautiful perceptual decision task that's been used with non-human primates, and we've been collaborating fairly closely with Josh Gold at UPenn. Basically, in this task, you have random moving dots. And then the task is to decide how many dots are moving in a certain direction when you add motion coherence. So the way you do that is there's the random noise in the background, and then you have either 10 or 20 or 30% of the dots moving in a particular direction. So what their decision is to let enough evidence accumulate that they're sure the dots are moving either to the right or the left. And so to make the task a little bit more interesting, because when dots are moving across a field, it's almost, if any of you are soccer fans, boy, people can move across that field like little moving dots. And sometimes it's chaos, and sometimes there's a pattern. So we'll use little vignettes or scenarios such that who's going to make the next goal, or who's most likely to make the next goal. Or for younger children, when he's been in that, trying to get honey again, so what direction should he run, right? And so that helps children understand it. And then we've used spaceship scenarios and this sort. But what we try to do with this task is relate one direction of motion to the 5 points and one direction to 1 point. And the way that we equate performance is we have an easy and hard condition, and we individualize performance so that we keep everybody in the easy condition at right around 92% accuracy, and everyone in the hard condition is right around 65%. And the data that I'm going to show you, the motion coherence in the difficult condition does not differ across ages. It's right around 10 to 20%. That's very few dots moving in a direction for you to be able to tell. And in the easy condition, it's about 40 to 50%, which is a lot easier to tell. Let's see if this gives you a better idea of the task. So they're moving dots, and if it's in one condition, you press if it's moving, sorry, I'm trying to get it for you. If it's moving left, it's your pointer finger, and if it's moving right, you use your middle finger. And now I can just show you a simplified version of the task, although it's a little bit more complicated with regard to timing. So they see a fixation, and then you see the moving dots. They've pressed because they thought the moving dots were moving to that side. A fish showed up, so that was one of the scenarios where we're feeding a fish. And then they see how many points they get for getting it right. So what would be the predictions for adolescents on a task like this? They're going to get more points on one direction. So do you think they'd be more biased to go in that direction more? We're equating performance. Are they going to be faster? We actually have paradoxical findings in the adolescents and the adults. What we see in the adults is this is an age by reward interaction. What we see in the adults, and these are z-scores, so anything below 0 means they're faster. The adults are much faster for the high reward, the 5 points, and they're slower for the 1 point, the low reward. But look what the adolescents are doing. They are slower when there's reward at stake, as if they're letting evidence accumulate to make sure they get that reward. Now, let's see how this plays out. Now, this is really a preferential bias, right? Both the adults are showing a bias, but they're faster to the reward. And the adolescents are showing a bias in terms of being slower and trying to be more accurate. But we've equated performance. We've also regressed out RT and the imaging data I'm going to show you. And basically, we see this ventral striatum area again is an area that shows a reward by age interaction. It's ever so slight, though. So we're seeing that adults are activating this region too, so I want to be very cautious in the interpretations in terms of the brain imaging data for now. We also see that the area that's modulated by reward are these frontal striatal loops. It's actually the basal ganglia thalamocortical loops you would expect to be involved when you're trying to make a forced choice like this. So the implications of this last study is that we're using very similar systems that in some cases can lead to suboptimal choices, but in others can lead to thoughtful, patient decisions on the part of the adolescent. So really, I think what the challenge for us is how do we use this insight in developing interventions that will prevent our youth from getting into harm's way. So if I can put this in another way, or other words, how do we help them pause and wait before they make that choice? And I'd love to talk about this more during the Q&A or even during the reception, because there's a few ideas and a lot of work that's being done there. So let's just come back and look at those misconceptions that the adolescent is impaired, can't make optimal decisions, has delayed prefrontal cortical development. I hope that some of the empirical evidence I provided today suggests to you that you can't really look at behavior with just thinking about one region. You have to think about the circuit, and you have to think about how regions within the circuit are talking to one another. So in the heat of the moment, it may be that these subcortical systems are going to win out over the prefrontal. So Kirk again wins out over Spock, and that's going to lead to suboptimal choices. But if you use incentive to change behavior, there are times in which you can get them to be even more thoughtful. And so they're going to use the prefrontal cortex, the rational system, or SPOC. But more importantly, hopefully we see that adolescence, by looking at it across development from childhood to adolescence to adulthood, that this is a very transient period of development. So when you talk about psychopathology and criminal behavior, and yet we see in the lab, too, that we're seeing the spike and then it goes down, is suggesting that it's a normal part of development and transient. And what it's probably serving, and this is really the punchline for David Dobbs' story, it's probably serving to get the adolescent in those situations so that they can learn how to regulate their behavior, they can learn what the rules are of the society that they are in. And so I think with that, I'm going to stop, and hopefully we'll have time for questions. Right.
So that's one of the things I think. And the other thing that I wonder if, and maybe you'll talk about this when your other talks on sensation seeking, because sometimes I wonder if it's not adaptive, not so much to learn what they're supposed to learn, but to create new things, to learn new possible actions for surviving better in a toxic environment.
So Dr. Buchheimer has just given little tidbits of the next two talks that I'm going to be giving on here. So in terms of this last bit that I was talking about with adolescence, the first question is about, is the emotional reactivity that we see in adolescence part of them sort of separating themselves and actually it being part of leaving the home nest, right? It's kind of disruptive and pushing them out. I see that in terms of their approach to incentives as well. The second part is the sensation part, which I'll talk about tomorrow with regard of individuals that don't seem to be able to delay gratification. That sensation seeking or that approach behavior, I think it absolutely is necessary for us to have certain mechanisms that sort of pull us out to explore, right? But why would we want to leave the safety of our nest when we're getting everything that we need there? But so something to attract us, and remember, those crushes during adolescence, oh my gosh, if you could get those, well, maybe not. That would probably be too disruptive. That's going to pull you out. But at the same time, the talk I'll give on Tuesday really talks about, yeah, you might get out there, but if a predator takes you out, the minute you get out there, you've done no better in procreating or going on and being an independent adult in whatever species you are. And so I'll spend some time trying to unpack why there's this heightened emotional sensitivity. You get really interesting responses in adolescence to cues of threat that you don't see in children and adults, very similar to the ones that you see in incentives. And so I think this is part of sort of the evolutionary perspective, and also with discussions with Barb Finley, one of these Evo-Devo people at Cornell, it's kind of been helping me think that way and think about adolescence as adaptive. Too often we forget about the context in which behavior is occurring, and that's very important, we're finding time and time again, to think about the context, the animal, the human, whatever the organism or the model. What is that context? And think about how, in that context, if they're emitting a behavior or expressing one, how is that adaptive? And if they're not expressing one, how might that be adaptive in the context? And so our mouse and human studies have really been exciting in thinking about those possibilities. Hey, wait a second. We can't go from wife to husband. Okay. So a couple of things on that. If you never experience a risk and nothing pulls you out of your home, you will never have the experiences to fine-tune a system to help you then know how to behave in those situations. In terms of the gender differences, you're absolutely right. The criminal activity, not so much in terms of psychopathology, though, too. But in terms of the risk-taking studies that we've done, we have not seen huge differences, particularly in this area in the ventral striatum. And unfortunately, it's also the case in terms of this peak in criminal activity, because I've been sitting on the juvenile justice reform, one of the IOM committees. Girls are getting more and more involved in criminal activity, too. So your points are really well taken. The gender, at least in the laboratory, is not quite as convincing as I thought it would be, too, right? Because we also know, if we take it back to psychopathologies, it is typically males that have the externalizing or impulse control type problems. They have typically more ADHD, conduct disorder, and the sort. And so we thought that we'd have a higher incidence or a gender difference there. But even when we've gotten enough power where we felt like we could tease it apart. Now, that's not true when we start talking about emotion regulation. Then we start seeing gender differences. And I don't want to say too much, because those are really interesting that I'll include in the talk. Yes? Jeff?
That picture at the top, are those the students or the Sackler staff?
So this is the Sackler Institute. And what I thought you were going to ask, because we were talking about earlier, Bob and I were talking about Rockefeller University, which is really what we're looking out on to, even though I'm at Weill Cornell, it's right across the street. But in this picture, you're seeing 4 or 5 faculty members and their postdocs and staff. And then anybody that has worked with me, I've tried to include in the project. And I had to, of course, get in my teenage son, too, when he was younger. What's over to the right is a postdoc and pre-doc of mine when they were going to a brain imaging meeting. Lo and behold, somehow their flight got delayed in Dublin, which was not anywhere near Barcelona. But so they said they weren't going to make it to the meeting the first day. And then I get this picture of them with this brain, but this was at the Guinness storehouse in Dublin. But I think they just thought, well, if she sees the picture of the brain, she'll think that this was an appropriate layover. So.
Speaking of your son, do you have advice on how parents survive the adolescence?
So I actually, there are a couple of things. I think it's really funny because after Dave Dobbs' story, and he talked to several of us, Bea Luna, Larry Steinberg, myself, Jake Eade. Then NPR did a special on this, and I thought they were going to ask me questions about the brain, but no. They just wanted anecdotal stories about being a parent of a teenager, because I'd kind of mentioned that I was. But again, apologies. In the lecture on Tuesday, I'll be talking about emotional reactivity and how important it is for the adolescent to have opportunities to regulate their own emotions. And I know you hate to hear anecdotal stories and scientific presentations, but this one with my son was really quite compelling because it sort of mapped onto what we were doing at the lab at the time. He came home one day. He must have been, I don't know, not even quite 11. And he's a really good kid. And he came home and he started saying language that even I don't use on a regular basis. And I was just so stunned because that just wasn't who I knew he was, that I couldn't even say anything. It wasn't that I was trying to do the right thing, but I just couldn't even say anything. And so then he, of course, goes, all right, then don't even say anything. Like, that's the worst thing I can do. But then he goes to his room, and he cools off. And, of course, he's an adolescent, so he's hungry as all get out because he's come home from school. I'm still in the kitchen cooking dinner. There's a great smell coming up to his bedroom. So about an hour later, he comes down. He doesn't say anything to me. He doesn't even have eye contact with me. What he does is he comes up and he kind of rubs against me, says nothing, and I just go, tough day, huh? And he says, yeah. And then he puts a big smile on his mouth because then he thinks, I'm going to get fed anyway, even though I engaged in that. What I think was important for him, not that I knew any better, was for him to not associate that emotional reactivity with himself, but for him to try to regulate it, and then to realize he's got to regulate it if he wants to survive and be, and get his food. So I think many times we tend to jump, and that's only going to exacerbate things when they need to learn how to regulate. That's one example. There's
one question, yeah, back there.
right.
Could it be that they're focusing on resources that are related to that? So reputation and material resources. Could this tie into that? That's why it's key to
Yeah, I think that's a really good point. So if you're thinking about how are you going to establish yourself in society, the ways that peers can influence their behavior that Larry Steinberg has shown, and even in terms of monetary reward. But this point system, was a little bit concerning. I felt like we had sort of evened out the playing field because it's not really money. It's not nobody else is seeing their score. It's just points. But it still could very well be. There are really about three things that characterize adolescents in terms of their behavior, and one is this really this tendency to be more influenced by social pressures and peers, which I think you've really tapped into. The other is they really do seem to be sensitive to environmental cues, like nobody's business, whether it's threat or positive. The third is, and this is the point I was trying to get at, is they often don't think about the future consequences. They act now and they don't think. And part of that, of course, is going to be prefrontal circuitry, and we have a number of tasks that have begun to pick up on that, but part not. Oh, sorry.
So what would you have done if your son didn't react that way and the behavior continued?
If the behavior had continued, he would have been so grounded, and he wouldn't have had dinner, and you have to set limits. So, and I think that's now this is Larry Steinberg talking. You really have to set limits, and you have to monitor your kids. None of us want to nag our kids. None of us want to be the bad guy at that point, but you got to set limits. And they actually, all right, just one other quick, okay, more questions, and we'll talk later about another example. Okay. Is there any research on children growing up on a farm as opposed to in the city? And so your point is going to...
My point is maybe the competition, the difficulties, the experiences you have, pressure, etc. I grew up on a farm.
Right.
That's why I asked.
Yeah, but you weren't a hellion when you were an adolescent? I know I was, and I see my son in a big city is, so I'm trying to sort of see. The stressors are...
Well, the difference is, too, when you are acting out, the police pick you up and take you home.
Well, right.
There are all those.
Right. So there are definitely many more. Another argument has been, look, the reason why I always said that an adolescent is just, the terrible twos, only now they're mobile and they can get around, right? And so one of the sort of criticisms of this research, I think, too, has been, no, adolescence is just because they have so many more opportunities. But, you still set limits, so it's a combination of those. You got to get them opportunities if they're going to learn how to survive in the real world, though, too. Yeah, Alcino. Oh, sorry. This is...
In addition to looking at
So, there have been a number of studies that have begun to do that. We haven't ourselves done that yet. A study that we've just begun is, because I've been so taken by the juvenile justice reform, trying to understand why is it that teenagers approach threat and they do some of the craziest things. But usually what it is they might be on a joyride and then something goes wrong, the cops show up, and then they veer off and then they end up knocking over property or hurting someone, right? So now what we have is making choices about reward or fear when they think they're going to be punished, when they think not that they're going to get a shock, but they're going to hear this aversive sound that's a mixture between, it's like white noise, like the alarm clock, and when you're trying to get a sporting event and you're just out of range, that aversity. So that's one direction we're going. But there's some really wonderful work by, who would you say, Evelyn Crone and Monique Ernst, who's done a lot of work as well. There's a whole group. Yeah.
So development goes on beyond the teenage years.
Yes.
And then, in old age, there are dynamic changes in the prefrontal too. And I was fascinated by your finding about how reward interacts with impulse control. So do you see the same trends when there is a weakening of prefrontal function as we age in later years? Meaning, this is about constraining your models with functional imaging, biology, and so on. So placing it on a different place. And
so I think that's
drug abuse too weakens.
Right. So in fact, Bashara, I never say his name right, Bashara, was one of the first in addiction research to sort of suggest you get this imbalance between these two systems and competing. And you do see that later in life. I hate to put another plug for a talk. Tomorrow, I'm going to talk about another period in development that is kind of close to that midlife crisis around the 40s. And it's a longitudinal study of, are you guys familiar with the delay of gratification task? It's the marshmallow task. You tell the little 4-year-old, you can have one marshmallow now, or if you wait till I get back to. So now we've been looking at that cohort that was originally tested at Stanford 40 years later, and we've used some of our tasks in the scanner. And 40 years later, they can't stop themselves. And it's really, really interesting, because these guys are successful. And so it just blew me away that that's still there. And then you get on the other end, right? So there has recently been an RFA by the NIH to start looking, and there's a number of investigators that have been trying to look at impulse control. I don't know adding the incentives on as much, but...
Okay, I think we'll need to save the rest of the questions for reception. Indiana, where is reception out there? Out on the patio. Let's conclude with a hand.